Hi, my name is Jourdan Ewoldt, and I'm a PhD student studying biomedical engineering at Boston University, and I'm currently working in Professor Chris Chen's lab. My work is applied to Thrust Area 3 of CellMet ERC, which is tissue engineering. While CellMet's goals are to create a healthy, functional cardiac tissue patch that can be integrated to treat a damaged heart, it is also important to understand pathological changes that occur in the heart during disease so that we know what kind of microenvironments the patch may be implanted in, and also to avoid triggering any pathological signaling that is known to cause disease remodeling. Thus, my project is on disease modeling and studying the mechanism of a genetic cardiac disease called hypertrophic cardiomyopathy. In this presentation, I'm going to walk you through the composition and structure of healthy cardiac tissue, give an overview of important characteristics of the contractile cardiac cells called cardiomyocytes, and explain the process we use in CellMet to obtain cardiac cells, and later describe characteristics of one of the cell lines that we are studying. I will then go over some of the technologies we use to study the disease both in 2D and in 3D cardiac microtissues, and review the challenges and long-term expectations in this project. The main function of the heart is to pump blood throughout the body so that it can obtain all the needed oxygen, nutrients, and hormones it needs, while also collecting waste. To do so, the heart is made out of muscle. A lot of studies in the field focus specifically on studying the left ventricle. Tissue from the left ventricle is composed of three main cell types, including endothelial cells, shown here on the top, fibroblasts, and cardiomyocytes, shown here on the bottom. The main function of endothelial cells includes creating microvascular structures so that the heart tissue itself can obtain nutrients. Since nutrients could only diffuse about 300 microns, any tissue thicker needs to be vascularized, otherwise the cells will start to die. Endothelial cells also communicate with other cells in cardiac tissue and help regulate the organization, metabolism, and contraction of cardiomyocytes. They also help protect cardiomyocytes during injury by releasing paracrine factors to signal changes to the cardiomyocytes. Fibroblasts maintain and remodel the cardiac extracellular matrix, or ECM, which is the structural component of a tissue. Cardiomyocytes are the contractile cell type of the heart and control the rhythm and synchronization of the contraction. Immune cells, including macrophages, are also present in the heart, but we're not going to focus on those today. Cardiac tissue is composed of multiple ECM proteins. The main protein is collagen type 1 and also collagen type 3, which provide tensile strength and extensibility to the matrix. Elastin allows the tissue to have elasticity, meaning the heart retains its shape after contraction. Fibronectin allows for cells to bind to the ECM via their integrin receptors. Basement membrane components such as laminin, collagen 4, and proteoglycans also help anchor the tissue and play roles in sarcomere formation, remodeling, and regulation of electrophysiology. In the matrix, there are also growth factors, which are soluble factors that are involved in cell-to-cell signaling, and also MMPs, which are enzymes that degrade ECM proteins during remodeling. In CellMet, we mainly focus on cardiomyocytes, although all parts of cardiac tissue are important to understand tissue structure and function. Cardiomyocytes are the contractile part of the heart. As an action potential travels between sarcomeres, it activates L-type calcium ion channels located in invaginations called T-tubules, causing an influx of calcium ions. This influx activates ryanodine receptors of the sarcoplasmic reticular, or the SR membrane, which then causes calcium to be released into the cytosol. This is called calcium-induced calcium release. This then causes a significant increase in the cytosolic calcium levels that can then activate sarcomere contraction. Calcium then binds to a protein in the sarcomere called cardiac troponin C, which then moves the whole troponin complex away from the actin binding site. This allows myosin to bind with the actin. The myosin head binds to ATP, pulls the actin filaments, releases a phosphate group to leave ATP, and then contracts the muscle. The SR then removes intracellular calcium, the troponin complex returns, The actin site becomes inactive, myosin binds to ATP, releases from the actin, and the muscle relaxes. Since adult cardiomyocytes use a lot of energy for all of this continuous contraction, they need to produce a lot of ATP, and thus have lots of mitochondria. Adult cardiomyocytes do not survive long ex vivo, and the cardiomyocytes have limited proliferation. So the use of human cardiomyocytes in research is very limited. Cardiomyocytes from animals such as mice and rats are often used to study cardiomyocytes, but there are some differences in sarcomere biology compared to humans that can impact our findings. Thus, deriving cardiomyocytes from stem cells has become a very promising strategy to study and engineer human cardiac tissue. Pluripotent stem cells are cells that have the ability to become any cell in the 3 germ layers, which includes endoderm, mesoderm, and ectoderm. You can take adult somatic cells, like skin cells, treat them with a set of factors, and revert them back to become pluripotent. These are called induced pluripotent stem cells, and these cells have the ability to proliferate and can be extended for further studying. You can also use other factors to differentiate these induced pluripotent stem cells into specific cell types. In this case, on the left, we have induced pluripotent stem cells on day 0. We let them become confluent and then add a molecule to activate the Wnt pathway. This allows the cells to become mesoderm. You can see this in day 1. Then we inhibit the Wnt pathway a few days later to direct the cells to become cardiac, shown in day 5. We then add insulin, since cardiomyocytes can metabolize it, and the cardiomyocytes start to form, which you can see in day 7 and 9 in the clusters. However, you can see that not all cells have the same morphology, and that there are some cells not within these clusters that aren't connecting with the other cells. This is because in differentiation, you don't get 100% cardiomyocytes, you get some impure populations of other cell types. We select for cardiomyocytes specifically using metabolic selection. So what we do is we remove all glucose from the medium and we add lactate, because cardiomyocytes can survive on lactate, whereas the non-cardiomyocytes cannot. So the non-cardiomyocytes will start to die off, leaving you with a more pure culture, which you can see in day 13. Using CRISPR-Cas9 gene editing technology, we can edit the stem cells to have mutations specifically known to cause different cardiac diseases and compare the effects of the mutation to cells with the same exact genetic background. In this project, we are studying hypertrophic cardiomyopathy, or HCM, which is a mutation in the gene encoding for myosin, the contractile sarcomere protein. HCM affects 1 in 500 of the population. On the left is a healthy heart, and on the right is one from a patient with HCM. HCM results in cardiomyocyte enlargement, myofibril disarray, hyperdynamic activity of the heart, thickening of the ventricular wall, seen here, which then leads to diastolic dysfunction since the ventricle cannot fully fill. HCM can also lead to fibrosis and arrhythmias, all of which can lead to sudden cardiac death in otherwise healthy patients.
Using a set of 2D assays, we set out to recapitulate some of the disease phenotypes in the edited cell lines. On the top, we stained the membrane of the cardiomyocytes to quantify the cell area, and found that the HCM cells had a larger cell area as seen clinically. A few years ago, the Simon Lab and Thrust Area 3 created a cell line using CRISPR to add a green fluorescent protein tag onto one of the sarcomere proteins called titin. This means that the sarcomeres are fluorescent, so we can track the sarcomere movement by acquiring videos of live cells, shown on the bottom left. Then, using a sarcomere analysis software developed by the Simon Lab called SarcTrack, you can track these sarcomeres over time. A metric, sarcomeric shortening, is a measure of contractility, and Christopher, who is in Thrust Area 3, found that the HCM cells had increased contractility, modeling the hyperdynamic activity seen clinically in HCM.
We next sought out to study and model the disease phenotype in 3D using our tissue-engineered constructs. First, we differentiated the cells into cardiomyocytes. We then mixed them in an ECM mixture with stromal cells, or fibroblasts, and seed them into a device shown on the bottom left. Within this device, there are 6 sets of tissues that can be formed. The cell ECM mixture is added to each of the wells, and then over time, the cells compact around the 2 pillars within the well and form a tissue. Then, around day 7 to day 14, we take videos of the tissues contracting, as you can see on the bottom middle, and using software, we can quantify the deflection of the pillars and quantify the contractile force and stress of the tissue. On the top, we have a wild-type healthy tissue, and the bottom is a tissue using HCM mutated cells. We have found by making both healthy wild-type cell lines and also HCM cardiac microtissues that there is increased stress or force production in the HCM cardiac microtissues. This again demonstrates the clinical phenotype of hyperdynamic activity. We plan to further study the disease in vitro with these models. One of the major challenges we face in this project is the maturity of the iPSC-derived cardiomyocytes. Currently, the derived cardiomyocytes are fetal-like in maturation. However, most disease occur in adult human heart. Thus, we may be missing or misrepresenting some of the disease phenotype since the immature cardiomyocytes do not have the same machinery as adult cardiomyocytes. However, there are efforts in the field and in CellMet to improve this. There are many approaches you can take, including stimulating the cells electrically, mechanically stretching them as they would be in a heart, increasing the substrate stiffness to better match a human heart, adding biochemical factors found in the heart during development, genetically altering cells to behave a certain way, or adding in other cell types that are found in the heart so that cells can communicate and mature together. One interesting study that was done by Casey in Thrust Area 3 is to form cardiac microtissues under tension and then electrically pace them early on, but increase the frequency over time to train the tissues. This method allows them to find increased maturation in many parts of their tissue, including tissue conduction, sarcomeric proteins, energetics, and calcium handling. Other issues are standardizations in differentiation, since there are many aspects to biological reagents that we use that can alter the cell's behavior. There are also many ways the platforms can be improved, but there's a balance between using your current platforms to study diseases now or improving your platform so that you can learn more later. This work uses technology and knowledge bases from CellMet, including the use of our 3D dynamic cell scaffolds for us to use those to study diseases in 3D, and also cell adhesion and scaffold chemistries so that we can study the interactions of cells with each other and with ECM protein. As a thrust in Thrust Area 3, we aim to use the technologies developed by CellMet in combination with our stem cell biology and tissue engineering knowledge to build a clinically useful human heart tissue. To do so, we aim to obtain an understanding of the tissue assembling processes and how to use new methods to control this assembly. I will now show you what some of the work in our lab looks like. Before we make our cardiac microtissues, first we have to prepare the devices. To prepare the devices, we have a mold that has the negative print of the device layout that I showed you on a previous slide, and we pour a polymer called PDMS mixed with its cross-linker, and the amount of the cross-linker impacts the stiffness of the PDMS after it polymerizes, and this can impact the tissue's function. So we pour the PDMS mixture with its cross-linker into the mold, and then bake it at 60 degrees overnight so that the PDMS can polymerize. And then after we take the device out, we cut them and add them to dishes, and this is what one device looks like with 6 wells for 6 tissues. We then put a series of treatments on the wells and go to the tissue culture room where we can seed the devices with cells and ECM. I'm going to just show you what these cells look like. So within the cardiac microtissues, we use 90% cardiomyocytes and 10% stromal cells, which are human mesenchymal stem cells in this case. So I'm showing you here what the HMSCs look like. And these are the more supporting cells. Now I'm going to look at the cardiomyocytes, which are the contractile cells of the heart. And these are the cells in 2D before we add them to the tissues. And you can watch them and see how they beat in 2D. We then remove the cells using enzymatic treatments, count them to make sure we have the right density, and add them to an ECM solution with different nutrients in the media. We then need to mix the ECM solution right before seating the devices, because otherwise the ECM solution will polymerize or gel, making it so we can't put it into the device. So here I am adding the ECM components to the cell mixture. I'm mixing it up to make sure it's evenly distributed. And then I'm going to add it to the 6 different wells of the device. I then mix it up and down once to make sure that the solution is evenly dispersed within the well. And then I'll flip over the device and rock it back and forth a little bit to make sure that the solution is evenly dispersed within the well again, and also to encourage the cell solution towards the top of the pillars. I'll show you what they look like. So over time, it'll start to compact away from the wells towards the center to the pillars. This is after 2 days, but after 3 or 4 days, it will be off of the wells and completely formed like the tissues you saw earlier. We then can study the contraction like I showed you earlier, but we can also look and stain these tissues for different proteins within the cells and proteins from the extracellular matrix or ECM. So this is me looking at some of the proteins and markers that I stained for that are important for my study. And you can then look at how these cells are behaving in 3D. So I'll show you what that looks like. So you can get a look at what our tissues look like. They're about 200 to 300 microns thick. And this is what some of the staining looks like in 3D. And this is our cardiac microtissue system.